Welcome to the 2017-2018 Wednesday afternoon lecture series here at the NIH. For those of you who don't know me, my name is Anirban Banerjee. I'm a tenure-track PI in the cell biology and neurobiology branch at NICHD, and I'm really presenting on behalf of the membrane protein interest group. Today we have one of the few named lectures in this series. So today is the NIH Director's Lecture, which means that the speaker was voted for by the directors of the NIH institutes. The reason I paused, it's not part of my written script here, because it is impossible not to feel overwhelmed when one even attempts to introduce today's speaker. And it's not really only because after performing trailblazing experiments in the ion channel field as a young faculty at Harvard Medical School, he decided that he was not satisfied with the resolution of the experiments that the then studies were providing him, and decided to crystallize potassium channels, which was at that point of time considered an impossible feat by many established crystallographers. And not only because after receiving the Nobel Prize at a very young age, he continued to push the frontiers of the ion channel and the membrane protein research field by solving the first structure of a eukaryotic potassium channel in 2005. Incidentally, the first eukaryotic membrane protein structure to be solved through heterologous purification. And also not because at a stage in his career when others would have chosen to lead from the back, this is when I was a postdoc with him, he would regularly come to synchrotron trips, stay up till midnight, past midnight, process crystallographic data, and then show up in the morning at 7 o'clock and continue to work on it. And not only because as late as in 2012, when the structure of the first two-pore potassium channel came out, one figure was actually was from experiments done by Rod himself. Because like really the proverbial visually impaired men trying to gauge an elephant, these are really but snippets of Rod's scientific persona. The hallmark of which is really his deeply passionate, untiring, single-minded, and singularly insightful pursuit of the problem that is the love of his life, how ion channels work. That is both exemplary and inspirational. I will not attempt to go through his awards because they are not really good descriptors of Rod. I'll just share two experiences. Right after I came to the NIH, a postdoc from Sriram Subramaniam's lab exclaimed to me, after coming back from a cryo-EM workshop, I met Rod there. And I think he was both surprised and shocked, and I was not, because in his own words that was published in an interview in Nature, he likes to be at the steepest slope of the learning curve, which was referring to the latest breakthroughs in cryo-EM that has really revolutionized both structural biology and the ion channel field, the latter much through Rod's own work. When I first came to Rod's lab as a postdoc, I was surprised how small their seminar room was for a lab of that stature. And I quickly realized that what that did was it focused everyone's attention onto the screen. And almost every group meeting, we would have agitated discussions that occasionally ended with Rod's voice slowly rising over the rest of us. And as everyone else quieted down, Rod would share, almost as a soliloquy, his insights on the issue. Personally, these are some of the deepest moments of learning of my life that I often yearn to get back to. We are lucky here at the NIH. Rod turns down way more invitations than he accepts. Without taking any more time away from him, thank you for coming to the NIH, Rod.
Thank you very much for that more than kind introduction, Anirban, and thank you for those who invited me here. It's an honor to be here and talk about some of the work from my lab. Yeah, and also in terms of, advances that my lab has been able to make, it's a very good example of you know, how the whole enterprise of science is amazing, because in a way, my whole career have been basically a parasite of great technological advances that others have made, both in the development of electrophysiological methods, the development of crystallographic methods, and now more recently, those who developed the hardware and software behind the new revolution in structural biology that has enabled cryo-electron microscopy to be the technique of choice for atomic structure now. So it's just really great to be in a society and in a world where we can pursue science with so much at our hands. So I'm going to talk about the subject I love, and that is the biophysics and biology of potassium channels. And I'll talk about that with just two examples of projects we've worked on in the lab over the last few years. And first, though, I'll give a brief introduction to a particular kind of ion channel, the potassium channels. And this slide is a cartoon picture of a cell membrane and nucleus. And in the membrane, I show a sodium-potassium ATPase, the important pump that concentrates potassium inside a cell and sodium outside the cell. And then I show a variety of ion channels that are passive conduits for ions to cross the membrane. On the top here, I show potassium channels and say they're hyperpolarizing. And what that means is when potassium channels are open, as some are at rest in most cell membranes, potassium runs down its concentration gradient until it charges them, leaves a negative charge, net negative charge behind, and we say charges the capacitance of the cell membrane, so that the voltage on the inside, the voltage across the membrane, balances the chemical driving force out. So that says to be the Nernst potential for potassium, and that's the origin of the resting membrane potential in most cells. Then you can have another channel, like a sodium channel. Of course, when it opens, sodium rushes down its electrochemical gradient, causing a depolarization or a switch in the charge across the capacitance of the membrane. But that process is always stopped then by something like a sodium-activated potassium channel. The sodium entering will open additional potassium channels and restore the membrane potential. So potassium channels set the resting potential, and they tend to stop excitatory processes. They basically put a gate on it, and so stop excitation. The potassium channel family is very large, so there are approximately 80 members of potassium channels in us. And this is just to give some idea of their biological roles, not to say it in detail, but there's a class here called inward rectifiers, and they do many things. They regulate the rhythm of the heart. They're important in neuronal excitation, important in secretion of hormones such as insulin. Then there are channels called slow channels. They're potassium activated, calcium activated, or sodium activated potassium channels that are important in a number of functions, including muscle contraction, auditory signal processing, neural excitability. Two large classes of voltage-dependent ion, large branches of the family called voltage-dependent ion channels that are important in various electrical signaling in the nervous system and outside the nervous system. And then a fairly mysterious class called K2P channels that we still don't know very much about. These channels, what makes them a common family is they all have a structural element called the selectivity filter that looks like this. I won't talk about it other than to say it is what makes them all members of the same family. It allows potassium to conduct across the membrane and sodium not to conduct. But if you look at structures of this family, you look at them from far away and you realize that outside of that little element that makes the selectivity filter, you can see that they're a morphologically very diverse family, ranging from very little small ones here that look like this to really big ones up here, and they're just a very varied family. So what's going on here, and what's our current understanding of why this is such a broad family? it's my way as a biophysicist of looking at this is that what's happened is I describe potassium channels as setting the membrane potential in regulating excitatory processes. And the thing here is that there are many ways that a cell wants to regulate an excitatory process. And this channel family has evolved to have many different members that respond to different environmental stimuli. There are ones I'd call chemical forces cause them to open, and this would be, a ligand-gated channel where here a ligand shown in red binds to the closed form and causes it to open. That might be a sodium ion or a calcium ion that comes in during excitation, and then the excitation is shut off by these ions binding to the channel and causing it to open. There are mechanical forces open certain potassium channels. So there are that means you push on the membrane and they open. And we're still trying to understand why that occurs and how that occurs. And then there are ion channels where I'd say electrical forces cause them to open, and those are the voltage-dependent ion channels. So in that first slide of the cell, I said that ion channels open, and the ions move down their chemical gradient and set the membrane voltage. But there are then channels that actually the value of the membrane voltage determines whether they're open, so they're recursive on themselves. And those are the kind of channels that make action potentials that are important in neurons for propagating signals long distances. So the idea is this family is very broad and diverse because there are members that respond to different environmental stimuli. And in a sense, you could say there's a basic pore and a selectivity filter, but then that was embellished with other structural elements that allowed that pore to respond to the different stimuli. So I'll talk about two members of this, and the first one is here. Two members of this family, just as examples. And the first one is called KIR3, or G-protein-gated inward rectifier potassium channel, or GIRK for short. And this kind of channel's role in biology is exhibited here. So there are two examples, and on the top of this slide, I'm showing a cardiac pacemaker cell isolated from the atrium, from the SA node region, from a mouse, put in a dish, and this is voltage and time, and these little black spikes are actually depolarizations. They're action potentials. And this kind of cell just spikes on a regular, very, very regular basis. And in fact, that's the kind of cell that sets the pace in the heart. But of course, our heart can speed up, our heart can slow down, and the slowing down of our heart is mediated by the stimulation of a certain kind of receptor. And so it's shown here that on this cell, if you add acetylcholine, which is the neurotransmitter released from the vagus nerve that slows our heart, you can see that not only does this cell slow, but it actually, this excitation slows, it actually stops here. And that's because this cell is isolated in a dish, and it's not under both the parasympathetic and sympathetic sides of the autonomic nervous system. So pure acetylcholine put on here causes a hyperpolarization, and only after it's removed, after a while, the excitations, the periodic excitations return. And here's a very similar phenomenon in a mouse hippocampal neuron. And in this case, it's not acetylcholine, it's baclofen, which is an agonist for the GABA receptor, G-protein coupled receptor. And here what's happening is this neuron is depolarized so that threshold is reached, meaning the potential at which this neuron will spontaneously spike. So it starts to spike, and then you come along with baclofen and you activate this receptor, G-protein coupled receptor, and it hyperpolarizes, the spiking stops. And in this case, instead of taking the baclofen away, you add an inhibitor of a particular potassium channel, the KIR3, or GIRK potassium channel, and the spiking returns. And that shows us that actually what the agonist is doing is by stimulating the G-protein coupled receptor, it's opening a potassium channel, hyperpolarizing the membrane below the threshold so that it no longer spikes. So we know a lot about this pathway in general in both the heart and the nervous system, a G protein coupled receptor is stimulated by a neurotransmitter, and when the GPCR is activated, it catalyzes the exchange of GDP to GTP on the alpha subunit. The G protein subunits are then released, and they come away from the GPCR and activate their targets. And if this is a GI-coupled receptor, the target, the second messenger is the beta-gamma subunit, and the target is the GIRK potassium channel. So this cartoon shows basically a schematic with structures of all the components. And what I want to address here is what happens to this potassium channel when it meets the beta-gamma subunit of the G protein. And then what happens when this comes over here, this activates the potassium channel until the alpha subunit has hydrolyzed the GTP back to GDP, at which point the alpha subunit gets high affinity for the beta-gamma, and by mass action removes it from the potassium channel. It diffuses around it, then engages a GPCR, and is again ready to be activated. So what I want to focus on in the first part of the talk is the process of how the beta-gamma activates the potassium channel. There are 3 ligands shown here. PIP2, called a signaling lipid, and almost all members of this potassium channel family are dependent in an obligatory way on the presence of that. Then I show beta-gamma, the G protein subunits that come always together as a single complex, and sodium ions. And these are the 2 ligands that I'm going to talk about and how they seem to act on the potassium channel. So here is a picture of the GIRK potassium channel. It's a crystal structure. This part goes through the membrane. This would be outside the cell, and this would be inside the cell, and this projects into the cell. This has PIP2 present, but not beta-gamma, and it is closed. And this red lining is showing the lining of the pore. So it's a crystal structure in the absence of beta-gamma, and it's closed. And here is the crystal structure of the potassium channel in the presence of the beta-gamma subunits. And what you see again is the same potassium channel. This is the part through the membrane. Here is PIP2. In magenta are shown sodium ions. And here are the beta-gamma subunits. And the potassium channel is a tetramer with 4 subunits. And you see that each subunit, or actually at the interface between each subunit, there's a beta-gamma G protein unit bound to it, 4 of them. So a 4-to-1 stoichiometry with the potassium channel. A very beautiful structure, and in fact quite remarkable that it could have been solved in a crystal for details that I won't get into now. It was a many-year effort, and then finally accomplished by Matt Wharton, who was a postdoc in the lab at the time and is now runs his own lab at the Volum Institute in Portland, Oregon. And so the G proteins bind, and what I want to address is, functionally, how does this open the channel? How many do you need? Is 1 enough? Do you really need 4? What's the affinity? And what is sodium doing? And these are very simple questions, and you might wonder why we don't know the answer until now, but it'll become apparent why this was not an easy thing to address. Because in a sense, all I'm saying is, okay, we got a protein that has an activity, and we have ligands that activate it. Why can't you just titrate it? Why don't we know the answer to this? But it's a tricky thing, because notice that the beta-gamma subunits are bound, so the top of them are up against the membrane, and it turns out the gamma subunit is lipidated. So really, these are membrane proteins, and it's very hard to control or know their concentrations. So that's one of the problems with not knowing the affinity of these. And how do you get at that? So what happens when the beta-gamma binds is this. So here is showing a rotation of the cytoplasmic component that unwraps the helices here that make the gate. And so you can see a rotation of the part. It's going back and forth between the bound. This would be beta-gamma bound, not bound. And you'll see when beta-gamma binds, it causes a rotation that expands these helices towards the open conformation. So we can see mechanically that the channel opens. But how many, again, how many beta does it take? Is one enough? Do you need 4? Do they bind independently? With what affinity? So how do you get at that? So the system I'll describe is a synthetic lipid membrane system. The system that scientists have used to study this process for several decades are cells, either whole cells electrically or excising membrane patches. And the good thing, of course, about whole cells is everything you need for the proper response is there. So if you study a cardiac pacemaker cell, it's got everything it needs. But you don't know what's in there, and you don't know how much of the components are in there. So in a synthetic lipid system, membrane system, you have 2 chambers in a little partition, and on that partition, you paint a lipid membrane, and then you reconstitute the components in there. So you know what lipids are there, they're synthetic or isolated, you know every component, you add the membrane proteins, you add the G proteins, you add the PIP2, and you know how much of what you put in there. And here's an example, for example, of an experiment you can do in this synthetic system that you can't do in a cell, and that is you make membranes with different mole fractions of PIP2 in them, and you can look at how PIP2 will activate this channel. That's in the presence of G proteins and other necessary components, but you can actually look at the PIP2 dependence. Here you can show that if you have channels in the membrane and you add the alpha G protein subunit bound to GTP, after a stirring artifact, you get no activation of the potassium current. But if you add the beta-gamma subunits after the stirring artifact, you see the potassium channels turn on. So this is a very nice controlled system. They turn on until you then apply alpha GDP, and then you can shut them off, just like happens in a cell, because the alpha GDP now has high affinity for the beta-gamma subunit and competes it away from the channel. Now, so that's how you do this, and in fact, with this kind of system, you could look at the sodium dependence of activation, and you could look at the PIP2 dependence in this way. But actually, the beta-gamma dependence takes an extra trick, and that's what I want to show you. And the problem is, you can make a membrane with a known mole fraction of PIP2 because you mix the lipids together. But you can't do the same thing with the beta-gamma, because the beta-gamma is a protein, and you can't, and when you make the membrane, you actually have lipid indecane, and you paint it onto it's an organic mixture, which you can't add a protein to. So the problem then with the beta-gamma subunit is to work, it has to be lipidated and bound to the membrane. Itself, it's insoluble. So you can't controllably put a known amount of beta-gamma in the membrane. But so here's the way you get around this. Instead, you make a lipid membrane with nickel NTA lipid at a known mole fraction, so you can do that in an organic mixture and make the membrane. And now you take your beta-gamma subunit, you replace the lipid group with a his tag, and that will then bind to the nickel NTA head group, in other words, stick to the membrane. And so you can then control the concentration of beta-gamma in the membrane. And here's what the experiment looks like. So here is current and time. And in this experiment, in the black traces, the nickel NTA lipid mole fraction is 0. And what happens is when you add, so the current level is 0, you add 2 micromolars, what I'm calling soluble his-10 beta-gamma, and you get essentially no current, a little noise, but now you saturate with the same membrane with lipidated beta-gamma, and you know you had channels in the membrane, but are not activated by the soluble beta-gamma. Now you repeat this experiment with a new membrane with a certain mole fraction of nickel NTA lipids, so that's about 2 in every 2 lipid molecules, mole fraction 0.0019, so 2 in 10,000 lipid molecules have a nickel NTA, 2 in 1,000 have a nickel NTA lipid head group, and so this time you come along with your 2 micromolar beta-gamma, and you get amount of current, and now you saturate to fully, you now put lipidated beta gamma in the membrane to fully activate the channels, and then you say, okay, about 0.6 of the maximum activation occurred under that condition. And so that's how you do this experiment. But you look at this and you say, well, wait a minute. How do you know whether you got this level because you didn't have enough nickel NTA lipid in the membrane, or because you had enough but you didn't fully saturate the nickel NTA with the soluble beta gamma? In other words, there are two equilibria here, one between the lipid and the soluble beta gamma, and the other between the tethered beta gamma in the channel. Notice that the beta gamma in solution does not activate the channel directly. But you can get the answer to that quickly by the following experiment. So this is showing this normalized current as a function of soluble beta gamma at the known lipid mole fraction. And what you can see is that it saturates at a value of a little bit above 0.6, and it doesn't go to 1. And why doesn't it go to 1? Well, it's saturated, so it must be that you fully saturated the lipid in the membrane, but you didn't have enough of them in the membrane to fully activate the channel. And you can then test that by, instead of having a fixed mole fraction of lipid and increasing the soluble beta gamma, instead what you do is you have a fixed soluble concentration and a fixed soluble concentration, and increase make different membranes with different mole fractions. And here you can see the activation is asymptotic to 1, just as you'd expect. If you now get enough of the saturated lipid in the membrane, you'll activate the channel. And in fact, you can now take these two graphs and make another graph. You can say, well, I have this relationship and this relationship, so let me plot this axis, this x-axis against this axis. And what I'm going to look at is actually the saturation of the lipid in the membrane. So this is actually basically a Langmuir isotherm showing the saturation of lipid in the membrane with the beta gamma. And so what it tells you is if you have this his-10 his tags and 2 micromolar concentration of soluble beta gamma, you'll fully saturate all the lipids in the membrane. So there's a little funny result that I'll show you that actually has a quantitative interpretation that is important. What happens if I do the same experiment with a his-4 tag instead of a his-10 tag? You get what looks like a very funny result at first. With his-4 beta gamma, you now see that in fact you're asymptotic to a higher value, but the affinity is lower. You can see what's going on if you do this same kind of analysis here. Instead, we now make the graph, the equivalent titration graph with a his-4. What you can see is when you're using a his-4 beta gamma, you get a lower affinity binding to this membrane, but the maximum value within error is 3 times what it is for the his-10. You get 3 times as much beta gamma in the membrane. And then you can realize what's happening by appealing to crystal structures of polyhistidine with nickel NTA groups. There's a rule that the nickel NTA group is coordinated by 2 histidines, and the 2 histidines have to be at least 1 histidine apart. So at least 1 in 3 position. And then you realize, aha, a his-10 can bind up to 3 nickel NTAs, and it does, whereas a his-4 can only bind 1. So you have a field of nickel NTAs, and when you come with his-4, they're lower affinity, but you can get 3 times as many in the membrane. When you use his-10, each his-10 tag on the beta gamma recruits 3 lipids, binds with high affinity. So what this all comes down to is the following. We know how much beta gamma we're putting on the membrane. We know if we use 2 micromolar his-10, you fully saturate the membrane, and you know the concentration on the membrane because it's the concentration of the 2-dimensional concentration of the his-10, 2-dimensional concentration of the nickel NTA lipid divided by 3, because each beta gamma uses up 3 lipids. So now with this background, we can get to the experiment I want to show you, and that is, here is a graph showing the normalized current, so activation of the channel as a function of the nickel NTA lipid and the sodium concentration. So remember these, I told you I wanted to get at these two ligands, and you'll see why. And I can't very well, I hope you can, but these sets of curves have a surface on them, and this surface corresponds to a model that I'll explain in a minute. Another representation of this same surface is slices along fixed concentration of sodium, shown here. So normalized current as a function of the NTA lipid mole fraction, which is just the unit I'm using for the beta gamma concentration on the membrane. And what you see is very steep activation curves that get shifted at different sodium concentrations, and I'll come back to that. So the model is the simplest imaginable model for this system. You have 4 subunits, and in the crystal structure, we see 4 beta gammas bound. And the model says, okay, let's allow 1, 2, 3, or 4 beta gammas bound, 1, 2, 3, 4 sodiums to bind independently if they want, but we can put in cooperativity terms to see if they're cooperative. And you ask, how many do you need? How many beta gammas do you need to open this potassium channel? How many are required, and what are their affinities? And the answer is, you need 4 beta gammas to open it. And the fits, you can't get them to work well, even if you say, let 3 open, at least 3. So you need 4. And moreover, they bind in a highly cooperative manner. So each one, the KD for each beta gamma, sequential beta gamma, is 0.3 times the KD. So the KD of the third beta gamma is 0.3 times the KD of the second, is 0.3 times the beta gamma of the first, which means the fourth binds with about 37 times higher affinity than the first. So they're highly cooperative with each other. So take these two things together, that you need, the fact that you need 4 and the fact that they're cooperative is the reason why you get these extremely steep activation curves. So you might look at that and say, okay, they activate, but realize this is almost square-shaped. And it's because of the high stoichiometry. And recall, even if there were not cooperativity, this would be sigmoidal, because if it requires 4, it's a power 4 relation. But on top of that, you have the cooperativity, which makes it almost a square step. And the significance of that will come when we look at, consider in a minute the effect of sodium and how it seems to shift these curves to the left and make them even steeper. But first, let me just give you an intuitive picture of this cooperativity. Recall that I showed you the structure of GIRK in the absence of beta gamma, and then in the presence of the 4 beta gamma with this morph that showed the conformational change that takes place by interpolating between the closed and the open structure. So here's a cartoon version of that. And here is sort of a closed GIRK channel and an open one. And in this picture, I'm very suggestive by showing beta gamma fitting well to the open version, but not to the closed. Now imagine if that's the case, in the fact that this happens as a rigid body rotation that we see in the crystal structure, then it would make sense that this is all or none. So once it rotates, all 4 binding sites are receptive, are a good fit. When it rotates back, they're not. So this simple picture gives rise to this very intuitive feel for the origin of the high cooperativity in this system. A comment about the sodium. Notice that with increased sodium, in fact, what happens is the maximums increase, and also the curves in the steep part shift to lower and lower beta gamma concentrations. Okay. What's happening in the model is sodium binding the presence of sodium causes beta gamma to bind with higher affinity, such that a channel with 4 sodiums bound, a beta gamma will bind 7 times higher affinity. So it's this cross-cooperativity between sodium and beta gamma, and that causes this shift. Now, the significance of sodium in this system is the following. This is an inhibitory signal. So when, for example, in the nervous system, GABA is released onto the GABA B receptor, it's activated, and it leads to opening of this potassium channel, which hyperpolarizes the neuron and silences it. When does sodium change in a neuron? A highly excited neuron has sodium entering as the basis of its excitation. And neurons with high excitability get higher sodium just under the membrane, particularly in the dendrites and in long extensions. But even it's documented well in the cell body. So a highly excited neuron has a high concentration of sodium, which would mean that the inhibitory input due to stimulation of the GABA B receptor will be stronger because of the shift in this curve. That's the idea. That's what we think is the biological significance of the sodium effect. It says that a more excited neuron will get more inhibition. So if you look at this, you say, well, how much more? Well, that would depend on where you are on this x-axis inside a cell when you stimulate the G protein coupled receptor. How much beta gamma is released onto the membrane by the GPCR. So, if you're out here, and suppose you're at a physiological internal concentration of 4 and you go up to 16, you'd say, okay, it increases, but that's not very substantial. You might have had a 20 or 30% increase in your potassium conductance. Maybe that will have an effect, maybe not. But if you're down here in the steep part, you can have a very, very large effect. So it really depends on where you are on this activation curve. In other words, this is generated in the lab in a bilayer system, but where are you in a cell? And then thinking about this made us realize, this is interesting. We can work backwards using these data maybe to figure out how much is generated inside a cell. And the experiment goes like this. And in fact, this experiment kind of came up while I was paddling with my friend and colleague Bruce Bean. I was telling him about these results, and he said, hey, that reminds me of something we've seen. And so here's the experiment that was carried out in Bruce Bean's lab by Keiko Ware, that when they looked at a bunch of neurons. These are substantia nigra neurons from the substantia nigra, dopamine neurons. They add baclofen to a set of neurons, and you can see this is current. You can see a hyperpolarization. Here is a case with 0 millimolar sodium in the pipette. So this is whole cell recording. You put the pipette on the cell, and the solution exchanges inside the cell. So in this case, it has no sodium in the pipette or inside the cell. Here is a case with 27 millimolar sodium chloride inside the pipette. And you can see the current now to the same amount of baclofen is much larger. It's a different cell. So they do a bunch of cells in 0 sodium and a bunch with 27 millimolar. And on average, you get a much larger current response in 27 millimolar. So then you take the curves from the bilayer experiments, the 32 and the 0, and of course 32 is supposed to be 27 millimolar, but that's the kind of mistake that happens when you communicate between New York and Boston, but it's close enough. And so what you do is you now generate a curve that is basically we call an amplification curve, where you divide this curve by this curve and get this curve, and then you ask, well, what's the fold effect and where do you fall here? And so you fall right here. It's about an 8-fold increase on average. It's a big distribution. Not all neurons are alike. Average 8-fold, and then you go down and you say, well, how much NTA lipid mole fraction does that correspond to? And then you can directly convert that into a G beta gamma concentration inside the cell. And I won't go into what that absolute concentration is right now, but I think the significance is it's very beautiful that notice that that is falling right in a very steep part of these curves. It's near the bottom of this one and near the top of this one. So as if the system has evolved to place the steep part of the G protein beta gamma activation curves such that changes in sodium will amplify the response, which then means that a neuron that's undergoing a lot of excitation, when it gets an inhibitory input, that input will be stronger due to the effect that sodium is having on the affinity of beta gamma for the channel. And so the conclusions from this part of the work is that the GIRK channel has four beta gammas bind, and they're highly cooperative, and this gives rise to this very steep dependence of channel activation on G beta gamma. Intracellular sodium concentration increases the affinity in the physiological range of sodium in the cell. GPCR stimulation generates G beta gamma concentrations that correspond to the steep G beta gamma dependence, so it's well matched. The above properties gives rise to this sodium amplification such that neuronal activity will make the inhibitory response much stronger. And then this I didn't really get into, but beta gamma, if you turn this into an affinity, it's quite low affinity. It's a high concentration that's generated. And this is consistent with the idea that as soon as the GPCR stimulus is turned off, and within the few seconds that it takes for the alpha subunit to hydrolyze GTP to GDP, the stimulus is then shut off on the potassium channel, and the potassium channels close. So it's a quick turnoff once the stimulus is removed. All right. So for the second thing I want to tell you about, it's a different channel. It's a different experiment, but it's kind of, you'll see that it has a conclusion that is very consistent with what we saw with the G protein gated channel. And you'll see what I mean by that, in that we see that the G protein gated channel seems to be a good switch that turns on very cooperatively. And look what happens in this case. So this is a kind of a new kind of, it's an experiment I can show you quickly. And it's one carried out by Rich Height. I should have mentioned that it was Weiwei Wang. Weiwei Wang is a postdoc who carried out almost all of the functional experiments I just talked about. And this experiment that I'll tell you about was carried out by Rich Height, who now runs his own lab at the Memorial Sloan Kettering. And you'll see the question we were after. It involves, the experiment was carried out with a channel called SLO2. And what you need to know is this is a channel that's activated by sodium. It's a neuronal potassium channel. It's activated by sodium. And the experiment we were interested in is this. So what I'm going to show you here is a set of pictures. So when an electrophysiologist records from a neuron or a cell with channels like this one in it, here's what you see. You see in this recording, this is current on the y-axis, time on the x-axis, and this is a sodium-activated potassium channel. It's in the absence of sodium. In sodium-activated potassium channel, absence of sodium, you see no channel activity. As you raise the sodium concentration, you start to see with some probability the channel open, and as you raise it further, you see more opening, and in fact, you realize that there are at least two channels in that membrane. So what the physiologist sees is fluctuations, the random hopping of the single molecule ion channels, and how you see the stochastic nature of opening and closing. And when we talk about a channel being open, we're not actually, we have in mind an open state, but actually the physiologist knows that that means there's some probability it will be opening and conducting. So we talk about open probability, and it looks like this, and you can calculate the open probability and make a graph, sodium concentration, open probability, and it looks like that for this channel. And now the question is, what's going on structurally under here? What do you see? And let's develop this, and I'll just say that electron microscopy enables this experiment, because in crystallography, you couldn't do it. In crystallography, you could do the kind of thing I just showed you. You could solve the structure of a channel in the absence of a ligand, you could solve it in the presence of a ligand, and you could get the two conformations, and you could show the kind of movie I showed you of a morph between the open and closed. But what does that mean in terms of the fluctuations, and what does that mean in terms of the number of conformations that might happen in between that closed and open state. So that's what we're after here. In crystallography, you couldn't do it because a good crystal requires an identity of what's in the unit cell, which generally implies an identity of structure under a given condition. Whereas in a cryo-EM experiment, you're actually taking your proteins spread out in solution, not touching each other, and in a sense, you should be able to get the structure, or all the structures that are present in the population of proteins. So in principle, you should be able to kind of do a titration of the structure, and that's what we're after here. And so the general strategy is to actually collect data under different concentrations of sodium and then solve the structures and use something called classification to ask what probability are open, what probability are closed, and what we were particularly interested in is how many structures do we see, and do we see intermediate steps on the way between closed and open? So that's what we're after. The way we did it is we combined our data sets, 4 of 5. We couldn't do all 5 because one of them, the 5th one, was collected on a different microscope, and you couldn't combine the data. But we liked the idea of collecting the data sets and then throwing them together and processing them together. That way, we wouldn't introduce any kind of bias in the way we process them. And then we process them together and classify and then ask, what data set did you come out of? We asked that later. First, let me show you a completely open and closed conformation of this channel. So here's the open version. I always show it as blue, and here's a closed version. And this shows the lining of the pore. And you can see there's a big conformational change that I'll show you right here in this movie. So this is, again, a morph between the closed conformation in the absence of sodium and then the open conformation in the presence of high sodium. Now, this movie did not come from the intermediates. I'm just showing you extremes right now, so you have a sense of what we're looking for. So you see it going back and forth between the closed and the open structure. You can see actually a mechanical expansion of this part in blue that actually opens up the helices that forms the gate of this channel inside the membrane. Those are the endpoints. So back to this titration. So what you do when you classify is you then, first in this case, little comment about this. In most cryo-EM structures you see, people are, often you'll see they'll start with a very large number of protein particles called, and then you might notice that the final structure presented represents a small fraction of the total particles. And that's because that small fraction was classified and gave rise to a best structure. That's perfectly okay to do. But in this case, we couldn't do that. We have to keep track of everything. We can't throw things out. The only culling happened here were things in the beginning that absolutely were pieces of ice, not particle. Then everything else has to be kept because we have to normalize for the total number of particles to get these numbers. One little comment that thing we have to be careful in cryo-EM is sometimes I see people referring to a conformation as being significant as a function of the conditions under which it was collected. But if you look and realize that it's only 10 or some small fraction of the total particles, that's not fair, because that was a subpopulation of particles, and you don't know really what the whole population is telling you. So that's something you have to be careful about. So with this classification method, you see in the first concentrations in the 4 data sets here, you can see that, in fact, if you ask for 10 classes, what you essentially find is one unique class. And the rest are all almost the same. So I'll come back to this. And in the higher concentrations separately, you find, again, 2 main classes. Well, you get 10 classes, but you compare them and you realize that there are only essentially 2 unique classes in here. So if you now go back and ask, so for the first 4 data sets, after this classification, you ask, okay, where did each class come from? So we asked for 10 classes, and I just told you there are only two unique ones. But right now, let's just keep the 10 separate and say, what happens as a function of sodium concentration? So you process them all together. Now you just make these graphs. And you say, OK, which particles came out? What's the fraction of class 3 at 20 millimolar, class 3 at 40 millimolar sodium, at 80 and 160? And notice that this class 3 is the only one changing in a significant way. It's growing. The rest of them are not doing anything. I mean, of course, as a net, they're dropping a little bit because they're going into class 3. But there's no systematic change of the red classes. And so what you see is this appearance of class 3. And if you then repeat over and over again, really starting at the beginning to look at the reproducibility or precision of this process, you can see that this is what class 3 does. Class 3 is always, these are 5 independent structure determinations, and you can see class 3 slowly grows as you increase the sodium concentration, whereas as examples, class 1 and 9 do not. Why did I pick 1 and 9? So it turns out class 3 is unique, and the other classes are all not class 3, and they're a little different from each other, only in that there's a rotation of the cytoplasmic domain with respect to the pore. 1 and 9 are most extreme. We can tell the difference between them, and we wanted to know if sodium affected the population, the density of their contributions. And the answer is no. They don't change as a function of sodium. So essentially what is happening here is if you make a graph like this, open probability, and now here open probability is actually open probability of the structure. And I've gone back and you can take out images and put red circles on the closed ones and blue circles on the open ones. And it's not that you can tell from looking at the image that they're open and closed. It's that after determining the structure and classifying, you know every particle that went into every class, and then you can ask, what image did that come from and where were you? And you can now put a color on it. Okay, so the color came from the post-processing, came from the classification. What you can see as sodium is increased, you get a conversion from closed to open. And what's very strange is we do not see intermediate structures, at least within our resolution ability, which is some, in the sort of mid-3 range here. And so, and you can see it's a large conformational change, so if something significant were happening in between, we should see it. We carried this out with fourfold symmetry, and we also analyzed it without imposing symmetry because we kind of assumed if there were intermediate states, it may be that one subunit goes at a time, but we saw no evidence of that. So it seems that it's, again, a highly cooperative process, and in fact, the Hill coefficient for this is about 3.5, something like that. And the subunits, there are 4 subunits here. And so it looks like as you increase sodium, what you see is you see a population of closed channels and then the appearance of open channels at some probability that grows as sodium concentration increases. And as hard as we look, so far we do not see intermediate states. So our explanation for this would be that actually, the low energy conformations would be the closed set that are very close to each other, just have slight rotations of the cytoplasmic domain, in the open state. In all the states, of course, it's not magic. It doesn't turn into one, but it means that the energy of all the intermediate states has to be higher than the energy of either the closed or the open. So there are relative wells, and it's higher in between, so it spends very little time in between, because essentially what we're trying to do is look at the Boltzmann distribution of structures as a function of sodium concentration. And what we see is only closed and only open, a highly concerted process. And in fact, if you compare the electrophysiology shown here in blue with the cryo-EM, and it only goes to 300 millimolar, It's kind of surprising how close these curves are to each other, and it's surprising for a few reasons. One is that, you know, when you, well, somebody might ask about that, but you'd think when you freeze or when you prepare the EM GIRK, there's actually evaporation. So do we really have the sodium concentration very well controlled? We don't know. But these are the data. There's one obvious difference. The black curve is clearly going to 1. I think if we went to 600 millimolar sodium, we'd be up close to 1. Here, we clearly are not going to 1. And so there is a discrepancy at the asymptote point of these. And what that discrepancy must mean is that within the set of channels that we define as open because they've undergone the large conformational change, there are small differences that we don't see that cause them not to conduct. And this is very common. Very few channels actually, functionally, even if you drive the stimulus all the way to open, very few of them go to an open probability of 1. Some do, but many do not. And so this could be as subtle as a blocking ion in the pore that we can't see at the current resolution, or a small kink somewhere, a small conformational difference. To me, it implies there are substrates within what we define as open that are subtly different. Some conduct and some do not, that account for this difference. So the summary from this part is that we see an ensemble of closed conformations, and they're sodium independent. They just fluctuate around and their relative populations of what we call closed, the red channels, do not change. The open conformation emerges in a sodium-dependent manner without stable intermediates. That is, the sodium-dependent gating is highly concerted. And again, as I mentioned, at higher resolution, in principle, we should be able to discern differences. And coming back to the question, do I really believe there are absolutely no intermediates? Well, of course there must be, but they're very low populated. And at least if ones are more highly populated, they're either very close to what we're calling closed or very close to what we're calling open, and not significantly different from either of those, so we don't discern them. And I just want to end with acknowledgments. The G-protein gated potassium channel structure work was carried out by Matt Wharton. Wei Wang did all of the functional data I've shown you. Koki Torihara studies a similar system in the heart. Wei studies the system in the nervous system. But I talked mostly about Wei's data. And our collaborators, Bruce Bean and Keiko Ware from Harvard, carried out the experiments on dopamine neurons. And the slow 2 potassium channel titration was carried out by Rich Height. Thanks very much.
Rod,
yes.
though the sodium titrations are really beautiful, and it's amazing to see this sort of structural correlate of the concerted opening. I'm wondering if you were to sort of make a hypothesis as to what an intermediate might look like.
As to what?
A hypothesis as to what an intermediate might look like. If you were to suggest it had 1 open plus 3 closed, sure. Is it possible to make a sort of hypothetical model and then go back to the data set and see if that helps you bring out?
You could do that. So the question is, you know, actually, to restate the question, of course, intermediates have to occur because it gets from closed to open. So they do occur, but they're very rare, obviously, because we don't see them. So they're very, you know, underpopulated compared to what we do see because they're at a higher energy level, presumably. And then the question is, what must they look like? So it turns out if you try to open this thing, if you try to open one subunit at a time, you do run into some problems because, in a sense, the way the helices wrap around each other, it's hard to move one without moving all of them. Mm-hmm. So it seems that my guess is it would have to inch its way out. Not that one subunit can snap all the way, and then the second, then a third, maybe in some ordered fashion. I think it's got to be a diffusional process where they kind of all open at once, because in a sense, they bump into each other as they open.
Yeah, that's cool.
Yeah.
Thanks.
Rod, going back to the GIRK structure, you have this beautiful high-resolution data. Can you tell us anything about how sodium modulates the affinity of beta-gamma? And then also, might the same mechanism apply to the slow channel?
Yeah, so the way I think sodium works, I mean, sodium's pretty subtle. Sodium is bound, so what I'll say is this. So the answer is, I don't really know. But what I do know is sodium, recall that the cytoplasmic domain rotated and caused a twisting of the in a direction that kind of opens the helical bundle that makes the gate. Sodium is bound at that interface between the cytoplasmic domain and those helices. And it makes very specific what look like structural interactions. And in my view, sodium tightens up basically better transfers the rotation of the cytoplasmic domain to opening of the pore, opening of the helices that make the gate. So I think that's what's happening given where it sits. Yes. And then how does that connect to the slow 2? And could the sodium be having a similar effect? And I don't think so. And it's because, so as hard as we have looked for where sodium binds in slow 2, we can't see it, which is a little bit surprising for the following reason. We've also determined structures at quite good resolution of slow 1 in closed and open conformations. That is a cousin of slow 2, but calcium is the activating ligand instead of sodium. And there we see where calcium binds very clearly. 2 sites per subunit, 8 altogether. If we look in the corresponding sites, we actually see density that we thought was sodium in one of them, what we call the RCK2 site. But when we mutate it, to make it go away, we still get sodium activation. So we don't know, and I think sodium works. Notice it was a very low affinity. It's a lot of sodium opens that channel. It takes a lot. So I think it's probably multiple low affinity sites, and it's why we don't see them. But it's working on this gating ring structure that kind of has this very specialized motion that is almost the same in the calcium and the sodium activated one, but is very different than the rotation that happens in the GIRK. Yeah. Yes.
Thank you for a wonderful talk. I was wondering in the GIRK channel, when you were referring to PIP2, are you talking about PIP452 or in any other variant?
I'm talking, so it's a good question. We've looked at a lot of substituted PIPs, and they have many of them activate. But it's really interesting because many of them activate with not only different affinities, but to different maximum values. It's a whole kind of bunch of data we have not published. But the one we're talking about here is PIP45. But the other PIPs have interesting effects, and in fact, some are competitive on this one. They bind but don't activate. And you can only study it by mixing them in that way to see it. And it must have to do with regulation in a way no one knows right now. Yeah.
Does it share any homology with any other PIP2 binding proteins? Or, for example, like pH domains and things like that?
Does what?
Typically, PIP2 binds to pH domains. So
yes.
Does it have similar structure like that?
The structure is quite different. The PIP binding site's quite different in the GIRK channels. So in a few of them, we have very nice structures of PIP2 bound, like this one, and in one called KAR2. And it's a different mode of binding. Yeah.
Thank you.